All right, so we're going to go ahead and get started. Now, what we're going to do is parts of blood. We're breaking down these test tubes are going to be, they're not even test tubes actually, they're called Vacutainers. So one of the reasons, if you guys can see my screen or where I'm presenting from, you guys are going to see there's this little thing called a Vacutainer, and this is where the blood goes. So we are going to look at what happens when we collect blood in the clinic and why we do it. So we have two different types of tubes that we're going to look at today, and these are Vacutainers, or vacuettes is what it says on the top there. Pretty much what it means is that this container has a vacuum in it. So as soon as I put the needle in with the syringe, it sucks up the blood. So I don't even have to push the syringe. The blood will just automatically go in here because there's a vacuum. So it wants to pull in and make that pressure even. So these are Vacutainer tubes. So I'm going to collect blood and then I'm going to put it in these tubes. The first tube that we're going to look at is a purple top tube. All right, so I'm going to go ahead and use a combination of text boxes and drawings. When I have a purple top tube, so this right here is going to be my purple top. My purple top tube has this chemical in it called EDTA. Now, EDTA is the shorthand for what this chemical is. It's like super, super long. You can look up what EDTA stands for. What this chemical does is it prevents the blood from clotting. Okay, so this purple top, this chemical in it, this EDTA, is going to prevent my blood from clotting. That means that this is going to be a whole blood sample. Okay, so when we're looking at a purple top tube, we're looking at a whole blood sample. So that means everything that's in this tube is whole blood. So everything that's in this sample is whole blood. Okay. All right, so this whole thing is whole blood. It's pretty much what you look like right now. What is in a purple top tube is going to be what is flowing in your veins. It's pristine. Okay, so think of purple top as pristine. Nothing has happened to it. It is what it is in this tube as it is in your veins. All right. There's three layers after we spin this tube. So this is going to be after we spin. Okay, so this is what the sample is going to look like after we spin it. So what is going to happen is what we're showing you is going to be no clot, no blood clot. Whoop. I'm then going to spin in the blood. And I spin it in a centrifuge. So there's this machine to put blood to. It's kind of like if you guys have ever gone to a carnival and you've gone into the Gravitron, right? Or that one ride that you go along the sides and then they spin you and they drop the floor and then you're just still stuck. Yeah. So that's pretty much that's a human sized centrifuge machine. And what it does is it spins you so the inertia pulls all the heavy cells to the bottom of the tube and it levels out the density. This is why you get so sick when you're on a Gravitron. Pretty much what you do is you're spinning and causing all of the heavier cells in your body to go to the bottom side. So no clot, spin. And then it's going to give us, when I spin this whole blood without a clot in it, it gives me something called plasma. So it gives me plasma. So no clot, spin gives me plasma. And that's what this top layer is. I'm going to go ahead and color that yellow. Now, plasma really isn't yellow. Plasma is clear. Plasma is really clear. Because this is just the liquids. Okay. All right, go ahead and flash a thumbs up or plasma in the chat if you guys are ready. Flash a thumbs up or type plasma in the chat. Cool. All right. So the other two layers, we have this middle section right here. This is called a Buffy coat. And then the bottom, does anybody want to take a guess at what the bottom layer is going to be called? Here, I'll give it to you because this one's kind of hard. I'll have you guess on the next one. This is going to be our RBCs. Okay, so we've got plasma's the top, Buffy coat's the middle, and then RBCs is the bottom. So I'm going to go ahead and also color in the RBCs. Oh, I'm like, why am I using my mouse? I've got this. Okay. All right. And then this Buffy coat that's in the middle is white. It generally will be white. If it's not white, so this is white. It doesn't, right? I can't tell. If it's not white, it might actually be a yellowing color. And if it is yellow, then that means that you have a lot of fatty cells. And then if it's spotted with pink or red, then that means something else in your Buffy coat as well. So when you take your whole blood, you are looking, after you spin it, we're definitely looking to see what that Buffy coat's going to look like. You may also need this plasma. Okay. So in my plasma, I have 4 different things in my plasma. So 4 things make up my plasma. So what I'm going to do is I'm going to number. So I've got 4 different things that are going to make up my plasma. The first thing I have that makes up my plasma is going to be water. And water is something like 97% of what makes up that plasma. So most of your plasma is water. Second part is going to be proteins. The third are going to be electrolytes. It's like when you drink Gatorade. And the last component is going to be something called fibrinogen and clotting factors. Right, because I don't have a clot in this. I don't have a clot in this blood. There's no, right, no clot. So I still have the fibrinogen and the clotting factors, the things that make the blood turn kind of solid. So that's the fourth element. All right, once you get the 4 elements, I want you to either type which element you think is the coolest or just give us a thumbs up in the camera. Once you get the 4 elements, either type your favorite or thumbs up. Okay, cool. All right. So now the Buffy coat. Now the Buffy coat is called such because it's buffering, right? It's the buffering between the two layers. So the Buffy Coat, that's an easy way to remember that because it's the buffer between those two. So my Buffy Coat is going to have two components. Buffy Coat has two components. The first component is going to be my white blood cells. The second component are going to be my platelets. Now, does anybody want to guess what is going to be in the red blood cell layer? Really? No takers? That is going to be red blood cells. Because it was a trick question, but not a quick trick question. All right, so your red blood cells are just the bottom. So this is where when you watch that at Dove video, when you were talking about the PCV or the packed cell volume, that's what the packed cell volume is. It's this bottom one. So we'll spin this down because we might have a test that tells us that we need to look at the plasma. We might need the plasma for the test that we're running. Or what we're doing is we're putting this in a microhematocrit tube or a super, super tiny glass straw or plastic straw, super, super tiny. And then we spin it so we can get this packed cell volume. So that's what they're looking at when they look at that PCV. They're actually just counting the red blood cells in it. And you use that Buffy Coat to find where, like, to be able to use that chart. Yeah, absolutely. We should be done in the next like 6 minutes. All right, so whole blood, purple top tube, no clot, I spin it, I get plasma. Cool, so think purple plasma pristine, right? I don't clot the blood, so my blood is the same in my vein as it is in my purple top tube. So plasma pristine, purple top. When we're ready, flip to the next page and then give me a thumbs up. All right. So now we're going to look at this red top tube. The red top tube, if you can see, says Serum Separator tube. All right. So the red top tube is going to be my Serum Separator tube. Now, some of these are going to have like stripes on them. Some Serum Separator tubes might even have like a little gel at the bottom of them. It all kind of just depends. So with a Serum Separator tube, I'm going to allow the clot to form. Then I spin. Okay, so I'm going to allow my clot to form. Then I spin. So I'll come down to my bottom. So we're going to do clot, spin, then what am I going to get from this? Once I spin it, what do you think? Am I going to have plasma anymore? Or am I going to have something different? I'm going to have my serum. So I let it clot, I spin it, and then I'm going to get serum. So this top layer is going to be serum. This top layer is going to be serum. And this whole thing is going to be clotted blood. So versus the purple top was the whole blood, this is clotted blood. So now we've allowed a chemical change to happen. So we only have 2 layers in this one. So I've only got 2 layers. I have my serum up top. And then my bottom, I'm actually going to have a cellular component. And when I spin it, because I have this solid and I don't have a Buffy coat, it might be on an angle. It's not necessarily always going to be straight because I have that clot in there especially. Okay. So those are the 2 components of the clotted blood. And again, I allow it to clot, then I spin, then I get serum. Now I'm going to break this down. It's an interesting breakdown with the serum because I've allowed my blood to clot, right? So this is still going to be clear. It's still the fluid component. So my serum is going to be my plasma without my fibrinogen. Because remember, I allowed it to clot, so then I don't have my fibrinogen or my other clot factors because I let a clot happen. And my plasma is my water, my proteins. Actually, I'm going to try to write that. There we go. It's going to be water, proteins, and my electrolytes. Right, without my fibrinogen or other clot factors. Thumbs up when you're good there. So water, proteins, electrolytes. So it's your plasma without your fibrinogen is the serum. Again, some tests might say you need serum. Or some tests might be like, hey, your blood was horrible. We just need the serum to be spun out. So now we have our cellular components. What's going to be one of the first things in my cellular component?
Red blood cells.
My blood cells. Okay. So I'm going to have my RBCs. I'm going to have what other kind of blood cells? Right, my white blood cells. And my platelets. What am I missing though? What haven't I accounted for yet? What haven't I accounted for? It's something that I'm lacking in the plasma.
Fibrinogen.
My fibrinogen, yep. So the last component is going to be your fibrinogen. Because remember, I made a clot. So I'm going to have, my clot is going to be down here. My fibrinogen and my other clotting factors are going to be there because I allowed that clot to form. Okay, so that's why it's not up here anymore, because that clot formed, so it can't be part of my serum. So it has to be down in the cellular component.